5:31. Well, my time at the moment is 5:31. For some of you, it's 8:31. Um, I'm going to wait for one more minute because I see that we are having a lecture where you're thinking, where did I put that link? It's wonderful to have everybody back with us. Great feedback this week. Great course for everybody. Somebody said, I don't have your voice. This week, I sent the recording from last week, Tuesday's course, and I hope that everybody got it. So for those that could not join us last week, before we delve in, I'd like to let you know that all lectures will be recorded and you'll receive a copy of this recording in the next few days. And if you don't receive a copy, please message me and I'll send it to you. We also have pre-course reading material. Second, as well as the course reading material that I have for you. So if you haven't received it already, with pleasure, I'll send it to you again. These recordings will serve for both training and marketing purposes. And I'm Jade Edelstein, the marketing manager at Genix. Many of you have already interacted with me by email or LinkedIn, and we have our CMO, Raviv Yitzhaki. Due to the large number of participants, I've muted everyone, but please feel free to utilize the chat feature as needed. As you can see, I found a great comic I saw in AI. It's funny how we all turn to AI to answer questions about its own impact on our lives and business. Perhaps the real question should be, how can we keep up with AI's evolving role in shaping genomics? Food for thought. And maybe a future Google search. Also, did you know that this week is Medical Genetics Awareness Week? This is a yearly celebration of the vital work of clinical and laboratory geneticists, genetic counselors, genetic nurses, and physicians, the people producing the technology and software devoted to translating genes into health. So if you would like to mention this on your social media pages, use the hashtag medical genetics awareness. You'll learn today from two experts, Dr. Hagar Moshaked and Dr. Ali Tabish, who both have extensive experience as senior variant scientists. This course provides insights into the latest advancements in genomics and personalized medicine, along with practical skills in NGS data analysis, variant interpretation, and clinical reporting. Here is a copy of the updated schedule. There have been some changes made from feedback that we received from the course. I will send it to you all by email. Some quick housekeeping. The Genix course will run from the 5th of March to the 30th of April. You will notice that the times may have changed depending on where you are in the world because of the change over to daylight savings time. Please check on the mail from Zoom with the link confirmation, the times for your times in your area. It could be an hour ahead or before. Also, essential to attend all lectures, and please keep your mic muted. Use chat to ask questions through these lectures as questions or ideas come up for you. And again, I'll send you a recording before the next lecture. At the end of the lecture, we will have a live Q&A session with Hagar and Ali. And contact me, Jade, if you have any questions at all. And now it is over to Hagar for lesson number 2.
Hi, everyone. As you know, we are pre-recording the session, so enjoy, and we'll be here live for Q&A.
In class number 2 of our course for clinical analysis and interpretation. In the previous class, I gave an introduction to NGS for diagnosis of rare disorders. I've also described the bioinformatic pipelines that are required for the transition from the processing of a FASTQ file to a VCF file and an annotated VCF file up into a file that is ready to be analyzed. And I've described a case study, which was a research analysis of a novel human knockout, which is a loss of function of WBP4. In this class today, I'm going to describe furthermore variant annotations and variant filtering. This is the heart of NGS analysis, so this class is the most important one in terms of understanding the basics of clinical NGS analysis. A quick note about variant annotations. There are many of them, and I wouldn't be able to review all of them and describe all of them. So this specific class is aimed to give you tools to learn more and to read more about other annotation sources. We are going to focus on the most important and highly used ones. I'm also going to give a case study, and today is going to be a clinical analysis and the way that this is conducted in a clinical genetic lab. So without further ado, let's start. So annotation sources, first of all, what's annotation? So an annotation is a note or explanation. Now I'm just reading the basic definition. It's a note of explanation or comment that is added to a text or diagram. Well, in genetics, we also use this term, and I can add here or a variant. So any type of information, any pieces of data that I can add to annotate, to provide further information on a specific variant would be an annotation source. And there are different categories of variant annotations. This could be on a genomic and genetic level. And for example, we can describe dbSNP, which provides a registry for many different variants in the human genome. And this could be describing which gene the variant is found in. This is another level of annotation. The second category is clinical evidence. So any piece of clinical information that I could bring, I could add to a specific variant is considered to be clinical evidence. This could be, for example, ClinVar, which is a registry of submissions of variants in a clinical context, and I will describe this deeply in this course today, in this class. But it also could be on a gene level, which is OMIM, for example, that describe the known phenotypes in human of the specific gene, or PubMed, which could provide further clinical evidence from the literature, from scientific publications. It could also be a phenotyper. A phenotyper is a tool that we have in GenX that correlates genes with phenotypes based on many different data sources. And this to further enable the user or the analyst to know what is the correlation between a specific gene and a set of phenotypes. And we described them in the previous class, HPO terms, but not restricted to HPO terms. Another category of annotation sources could be effect and predictions, which are mainly in silico software or tools to try to predict the bioinformatic, from a bioinformatic point of view, what is the outcome of the prediction of a specific variant on the human genome, on the functioning of the human genome. There are many different prediction softwares out there. Just as an example, I gave you a few. Mutation Taster, to taste the variants, that's the meaning of the name. CADScor, which I'm going to elaborate more in this class. SIFT, Polyfin, and GERP, which provides an evolutionary conservation score. And I'm also going to describe more about this one. The fourth category of variant annotation is NOMAD, or allele frequencies. Among them, the highest our highest used one is GNOMED. We also have the UK Biobank, the DGV, and 1000 Genomes, and there are other projects out there like All of Us that provides access to the data for certain researchers and physicians. Allele frequencies are maybe one of the most important annotation source for a variant, and basically it means, have I ever seen this variant before, and in whom have I seen it? Was it a healthy individual? Was it an affected individual? And in most genetic labs, there is a local registry or a database that provides the cohort or the local population. This is maybe the most important allele frequencies because there is noise, there is specific ethnical variants that I have to normalize against. And this is a very important piece of information when analyzing the data. And this goes, the power of this growth with the numbers of the local databases. So as many more samples that you have locally, the better analysis that you would be able to provide the patients. So this is another time the annotation categories, and these are screenshots from the Genix platform. And as this course is a hands-on course, we would provide you in the next class some training on the tool itself. And for those of you who've opened a self account and would like to do some hands-on experience or to have hands-on experience, and they would be received with VCF files and patients' data, they would see the screen, and this is why I'm already providing here some information or screenshots from the software. So the annotation categories in Genix are exactly divided into these 4 categories, and we can see here the genomic and genetic data, in which we have the location, the reference variant, the alternative variants, and this is information that comes from the VCF file. And then the basic annotation of every variant is the gene that the variant is found in, and the AGVSC, which we described in the previous class. And this describes the variant itself and the effect that this variant predicts to have on the gene itself. And the zygosity, which also comes from the VCF. We always ask ourselves, is this a homozygous variant? Is this heterozygous or a hemizygous variant? And the zygosity is fundamental to the genetic analysis as it correlates with the inheritance that we suspect in, for example. The second category, as we said, it was clinical evidence, and we have the phenotyper that provides a score of the correlation between the specific gene and the phenotypes that the patient has. We also have ClinVar indirect, and we will describe all of this. We have OMIM and OMIM inheritance, and there are 2 tools to provide variant level publications. The first one is LitVar2. This is a great tool by NCBI. I encourage you to click on that bar and go to the actual external link or LitVar, and this would provide you publications on the specific variant. And also Mastermind, which is not publicly available, but if you have your own login information, you would be able to see and further investigate into the variants by publications. The third category is effect and prediction. As we said, these are in silico softwares or tools that tries to predict the outcome of a specific variant. We have the effect of the variant, and then a list of prediction softwares, and I'm not going to describe all of these tools. Some are considered better, some are considered less, and it really depends on the context. But overall, we found out that the combination of several ones is probably better than relying on a single one. And for that, we created the severity score. This is the second column after effect. The severity categorized variants into 3 levels: low, medium, and high. And with that, we try to simplify the combination of all different tools based on the variant effect. So for splicing variant, we would look on Splice AI, for example, and other splicing predictions. For missense variant, we would take the relevant prediction tools and we will combine them under a specific algorithms that we wrote in-house. We would try to, with this, we try to simplify the process of understanding whether this variant is predicted to cause or to be pathogenic, or it is predicted to be benign. The fourth category is frequency, as we described it. So we have NOMAD, we have different sources for allele frequencies, also dB/dB here, and there is a local allele frequency as well, which provides you the allele frequencies in your internal lab in Genix. When we talk about annotations, there are two levels of annotations, and this is really a high-level categorization of all annotation sources. Annotations could be on a gene level, or they could be on a variant level. And this is very important, but because while most of the annotations are on the variant level, gene annotations are also crucial for the analysis. And some examples of the gene level annotations are phenotypers, like phenotypes-based annotations like OMIM and the phenotyper that I've described, and there are more like GeneCC, etc. And there are also constraint matrices that are important, and they are on the gene level. We are going to describe this further along. That said, many of the data sources contain information both on gene and variant level. For example, ClinGen and ClinVar, we are going to describe these as well. But what's important here is to highlight that whenever you look on an annotation source, you should understand whether it's on the gene level or is it on the variant level. Now, after we annotate the VCF with different types of data sources, and this could get to dozens of data sources, there is a computational variant filtering that we need to perform as a next step. Because after, let's say, a whole genome sequencing, there are about 200,000 to 400,000 of annotated variants per one individual. And this obviously is a huge number, which we cannot really start to analyze manually. And so this is the step in which we do computational variant filtering using all the annotation sources that we performed, all the annotations data that we added to the variants. The first one is filtering using usually. And just to explain, this is a general scheme. Every lab, every genetic lab would have its own computational variant filtering approach. This is just something that I got online, and I personally take a different approach, but this is just to convey the idea of how do we handle the data and how do we use the annotation sources first to provide a computational variant filtering before we start to analyze it manually. So filtering using population frequency data would be the first layer, because if you have a variant that is found in 90% of the population, for example, in a healthy population, obviously you would suspect this is not a pathogenic variant and you would want to remove all these common variants. And for that reason, this should be the first step. After that, we filter based on the coding regions of the exons. So as a thumb rule, we take 85% of the variants that cause a disease that are pathogenic. The variant would be in a coding region. And for that reason, we usually would filter out the deep intronic variants, the promoter variants, the enhancer, if we cover them and we are talking about a whole genome sequencing. If this is an exome sequencing, we don't even sequence them to begin with. Now, when we talk about the intronic regions or the splicing regions, here it says that you filter everything that is outside the coding region, which is plus minus 2 base pairs from the splicing junctions. This is not recommended at all. As I said, this is just a random scheme from the web, but today we take about 30 base pairs into the splice region, and we define this region as a splicing region. And we take the entire thing and we annotate and analyze it further. And we can also filter out non-splice related synonymous alterations. So if we have a synonymous variant inside the exon sequencing, inside the coding regions, usually these would be harmless and we can filter them out. Of course, there are exceptions. So everything that we describe here is just a common procedure or approach to filter variants, but we have rare diseases and we have rare cases in which a deep intronic could be pathogenic, and also synonymous variants inside a coding region could be pathogenic. But as we take one approach to all our exomes or genome data, we have to filter the variant kind of aggressively, just to start beginning analyzing a fair number of variants. So usually we filter out all the synonymous alterations that are not on the splicing region. And then we can have a family inheritance-based detection. For example, if we're conducting here trio analysis and the parents are related, we would try to consider homozygous variants in the problem that both parents are heterozygous. So for example, any variant that is homozygous in both parents or in one of the parents, I could filter out. So any approach, so at this level, I would filter out variants that are not consistent with the hypothesized or with the hypothesized inheritance models for the specific trio. Same goes with the local database or with your local allele frequencies. Many times we have a lot of false positives here. There are sequence artifacts that we see again and again, and even though they could be potentially look pathogenic, they're not, because they're found in many of your samples from the in-house database. If you are conducting or going through all these steps and you are defining your filters properly, you should end up with about 50 to 300 variants that needs to go into a further analysis, a human curation, and the actual analysis. The filtering approaches, as I said, are different between one lab to another, but overall the principles are the same, and this is what I'm trying to teach today. So with that, I want to start reviewing some important sources, annotation sources. So starting with OMIM. OMIM is a gene-level annotation source. OMIM is an abbreviation of Online Mendelian Inheritance in Men, and it's an online catalog of human genes and genetic disorders. It's a comprehensive tool or database, and it contains a lot of information on genes in general, and on the phenotypes of genes. Not all genes they have phenotype, but OMIM provides two types of registrations. One is for genes, and we can call genes that have an OMIM submission like an OMIM gene. And the second layer is genes within OMIM that do have a phenotype that is described in the literature. OMIM is freely available and is updated on a daily basis, which is great. This is how the UI, the first screen, looks like. And you can search here a clinical feature, you can search for a gene or for a phenotype or even for a region. And if you type in a gene, for example, I've just provided a screenshot for a specific gene I've typed, which is AGBL1. This is a gene that is known to cause corneal dystrophy in the inheritance model, which is described in the first screen. OMIM is autosomal dominant. So OMIM is a great database and a source to understand more about genes and their phenotype, and a phenotype that is associated with these genes. It is updates frequently every day. They have a log in their website to show how many updates have they done. It's curated. It's human curated, so it does not contain a lot of garbage or unreliable information. And it, as I said, frequently updates. That said, many of the publications from the recent year are not going to be here in OMIM because it does take sometimes even 6 months, 7 months, 8 months for OMIM to be updated with novel phenotypes that are described in the literature. Just as an example, if you go today to OMIM and you try to type WBP4, which is the novel gene that we described in the previous class, you wouldn't find the phenotype here because it was just recently published. So there is a way in OMIM to submit like a request to add a novel gene, a novel phenotype, but still, although this was submitted, it does take some time for OMIM to be updated because they take this job seriously and they review every publication and they decide whether it's worth or whether it's mature enough to be integrated into OMIM. But we do have to remember or to understand the restrictions of every source. OMIM is not updated with the latest publications. And if you have a gene that you suspect at, do not settle or do not only check it in OMIM, you have to go to PubMed and search it actively to see whether there are novel publications from the last 6 months, for example. The next annotation source is ClinVar. This is the URL. You can just tap and go. Easy to find, obviously. And ClinVar is a freely accessible public archive that reports of relationships among human variations and phenotypes. It processes the submissions of reporting variants that found in patient samples. And it also provides the clinical significance and information about the submitter, for example, and other supporting data. When we talk about clinical significance, so we classify variants as one of 4 categories: benign, likely benign, variant of unknown significance, likely pathogenic, and pathogenic. This is the basic classification of variants. In ClinVar, you can classify variants in other categories as risk factor, as supported, and other types of classification that they decided upon. The basic categories, the 5 categories that I've just described, comes from the ACMG guidelines, and we are going to have classes aimed to learn more about the ACMG, so I'm not going to explain about this today. The alleles in ClinVar are described in submissions, and they're reported according to the AGBSC standards. So today in ClinVar, there is a standardized protocols of how to submit a variant, and it goes through a curation steps of the ClinVar team, and they reject many of the submissions of researchers and physicians if these are not curated enough or if there are missing information. ClinVar in the past used to contain a lot of, I would say, garbage or a lot of clinical significance variants. And this is a well-known phenomenon. You could not trust ClinVar as this is not curated internally in ClinVar, but it is based on submissions of researchers and physicians worldwide. We take ClinVar as a supporting information. We would not handle ClinVar or use ClinVar information as the one that determines the classification of the variants. And this is highly important. In every lab and every expert could describe a process in which a likely benign classified variant in ClinVar was actually a pathogenic one eventually. There is also an effort to try to clean the data of ClinVar, to remove old submissions, to curate them, to update them. But of course, there are thousands and hundreds of thousands of variants in ClinVar. This could take years until we curate every variant. This is how the UI of ClinVar looks like. So you can search for a gene, a variant, a condition. The search box of ClinVar is not that convenient. I guess that some of you may, if you had an experience, you could agree. It's a bit hard to find a specific variant that you aim for because they have a format, a specific format. It's not a flexible one. But if you go here to advanced, at the end, you could find your variant. Probably the easiest would just be to type the gene and get all the variants, and then you could really nicely filter based on different characteristics of variants in the gene. Now, one thing that is less common or known about ClinVar is the accessions number. In CleanVar, there are 3 types of accessions or record accessions numbers, and they have a pattern of 3 letters and then 9 numerals. And the letters are SCV, which means submitted record in CleanVar. And then there are RCVs, which are reference CleanVar record. And these are data aggregated by variant condition pair, and I'm going to describe it right now. And the third type of a CleanVar accession would be VCV, which is variation CleanVar record. So there are 3 levels of records and accession numbers in CleanVar. If I provide, for example, an example for an SCV, this would be a submission by a specific lab. So we can see here a variant. This first row here is a submission by a specific lab. Here it's the ClinGen committee, and there is an accession for that. So every time you, your colleagues, myself, we submit a variant, this receives or this gets an SCV, which is the submitted record in CleanVar. Now, for a single variant, there could be hundreds of submissions worldwide. Now, CleanVar aggregates all of these submissions by the variant condition pair. So when you submit a variant, you need to choose under which condition you're reporting the variant, because as we know, certain genes have more than one phenotype that is known in human. There could also be more than one inheritance model, for example, autosomal dominant and autosomal recessive. So because of this, when you submit a variant to CleanVar, you have to specify the condition for which you are reporting. Now, when you aggregate all of the conditions together, this would receive an RCV, which is a reference CleanVar record. This is how it looks like. So above the table of the submissions in CleanVar, there are conditions, and they are aggregated under germline, and they're aggregated under somatic as well. So this is the condition on the left side, and then there are the RCV, which is again letters of 3 and then 9 numerals. And then there is the VCV, which is the variation CleanVar record, which is the highest level of variant submission number, which is just the entire variant and under this all the submissions and all the variant condition records. So I hope that makes sense to you. I mean, it's really important to understand under what level of record you're working on. And the accessions are versions, this is also very important. So you see after the dot here, after the 9 numerals, there is a dot and then the version. As SCVs is the version of the increment when the submitter updates the record. So as a submitter, you're encouraged, if there is an update on the variant, you can update it. And then for every submission, if this is the first submission, you would see a .1. And if it was updated by the submitter, this would be .2, .3, etc. So by this, you can see if this variant was updated by the submitter, or is it the initial submission? RCVs and VCVs versions are incremented when the content of the record change. So if one of the submissions, for example, as a new version, then also the RCVs and the VCV would receive a newer version. And also, of course, if there is a if there is a new submission by a new submitter. So I hope I made CleanVar a bit more clear to you guys. And basically, CleanVar reports the level of review, which is also very important. So this is another layer of the submissions in CleanVar. So the review status is, you can see it here on the right-hand side. This is the review, is provided by stars, and this provides on the aggregated records, VCV and RCV, and not on the single submission variants. The review status provides us a certain level of validation of the submission. So if there are, for example, 3 stars, we can go here to this table and see that this was reviewed by an expert panel. If there are 2 stars for this submission, we can go here and see that this was criteria provided, and this was submitted by multiple submitters, not just one, and there are no conflicts. Now, no conflicts means that all the submitters reported or classified the variant in the same class with the same classification. If there is a 1, which is the lowest degree of review status, so it's either that there is no assertion criteria provided or no classification provided or no classification for the individual variant. So when you review a variant in ClinVar, you have the review status, the stars, that helps you understand whether this is a validated variant or maybe it's not that of a validated variant, and you can use the scoring method to assess the pathogenicity of your variant. Now, going from ClinVar, I want to switch to a new topic, which is evolutionary conservation. Now, as humans, we understand that during evolutions, many variations were inserted between different species, but important regions in the human genome, we call them highly conserved regions, are predicted or are considered to be more important for the basic functioning of cells, of enzymes, of proteins. And evolutionary conservation in general is the presence of a similar gene or a portion of these genes, or even a chromosome segments in different species. And this to reflect, or that thus is reflecting both the common origin of the species, and also it's important for the functional property of the conserved element. So although we are highly different or very different from rice or mice or monkeys, there is considered amount of genetic content that is being evolutionary conserved among all the species. Now, evolutionary conservation comes in different levels. The first one is on the chromosome segment. So we know that there are segments of chromosome that could be conserved between different species, not necessarily humans. We're talking about an entire species. And there could be on the gene level. So there are genes that are highly conserved among different species, and we would see them with the same genomic sequence among different species. And of course, there could be a conservation on the amino acid level. So we know that there are specific amino acids in the protein that are highly important for the function of the protein. And if we take a random protein, not all the amino acids would be conserved on the same level. Some would be higher with higher conservation, some would be with less conservation, and that depends, is it in which domain of the protein this amino acids is found in. And of course, there is a nucleotide-based conservation, evolutionary conservations, which dictates all of the above levels. One of the measurements, the most highly used measurements for genomic evolutionary conservation is the GERP or the GERP. The GERP is a genomic evolutionary rate profiling. And this is a method to produce position-specific estimates for evolutionary constraint using maximum likelihood of evolutionary rate estimations. And there are 2 types of GERP. I'm going to provide you more details about the ARAS, rejected substitution score, which is mostly used for GERP. And this is the number of substitutions that are expected under neutrality minus the number of substitutions that are actually observed at a specific position. So when a GERP was developed, they, for example, took a specific position in the human chromosome, and they went through all species to see what is the expected substitutions of the same position between all species, and then what is the actually observed rate of substitutions between all the same species. And the scores of the GERP ranges from -12 to approximately 6, with 6 being the most conserved positions. So if I'm looking on a specific position in the human chromosome and I go through all species during evolution, and we do that many times when we want to assess whether a specific amino acid is conserved. And we see that the same amino acid in human is also found in the rice. Then we say this is highly conserved, a highly conserved position. And this would receive probably about 5.5 or above score of the JERP. So this is just a link to the actual data source. This was a research effort that was published many years ago already, but it is still a commonly used method and a very important score when you want to assess a specific variant. Now, one thing to keep in mind when we talk about evolutionary conservation, if you have a severe phenotype in humans, let's say childhood onset severe syndrome, and you see a specific variant and you suspect that this could be the pathogenic variant in your patient, then obviously this variant should be in a highly evolutionary conserved position. But if you work on a mild condition, and if you work on an intellectual disability, for example, or autism condition, then we might expect that genes are important for the intellectual of the human would not be that conserved during evolution. Also, if we're talking about multifactorial or polygenic disorders or traits in the human genome, obviously these would be less conserved, and I expect them to be less conserved. So with all the annotation sources, it is very important to understand the limitations of every source and how to use them properly, because at the end of the day, this is what counts for the analysis, for the genetic analysis. So going from evolutionary conservation, there is an effort to try to generalize evolutionary conservation as one characteristic of a genomic region, and to try to predict whether a specific variant in a region in the human genome would be pathogenic or not. And I've just brought here some a cover from Science predicting pathogenicity. And this is about deep learning forecast of impacts of human missense variants. Great paper. I highly recommend you to have a further read on that. But this is just as a complementary information to this slide, in which I want to describe more about the pathogenicity scores. So we know that in silico pathogenicity prediction tools, there are algorithms that are designed to predict the functional impact of a variation. Usually these would be missense, but not only missense changes that are caused by single nucleotide variants. We do not yet have a tool that is being able to combine different variations together or look on a multivariant score, which is harder to provide. Now, the tools integrate a number of features in order to assess the impact of the variants on the protein function. First of all, whether the effect of a variant, for example, whether it's a non-synonymous or synonymous. This is very important to predict the outcome. And then what site of the if the site of the mutation is conserved or not, and that's where the JERP score gets in. But of course, not only, because there are other parameters of conservation. And then whether the mutation is able to change the structure, the secondary and tertiary structure of the protein or not. So we know that there are alpha and beta sheets, and these are alpha helix and beta sheets, and these are important for the function of the protein. Some mutations or some regions of the protein would be highly conserved, or the specific amino acid structure is very important to provide the further structure of the protein. And the mutation, or these prediction tools, would try to assess whether the specific mutation is able to change this structure or not. One of the parameters that we could also try to assess whether the variant affects an active site or not of the protein, or an important known domain of the protein, whether it could promote an alternative splicing. So for splicing variants, we have a dedicated prediction tools to try to assess whether these variations could affect or could cause an abnormal splicing of the mRNA. And also whether they are introduced an unexpected stop codon, which would lead to a loss of function. And we described the effects that cause loss of function in the previous class. And just as a short reminder, we are talking about stop gain, frame shifts, splice donor, and splice acceptor as the four types of effects that would predicted to cause loss of function. Now, one of the highly used and I would say most appreciated tools is CADScore, one of the prediction algorithms. And there is here the PubMed ID, and you're welcome to have a further read on that. And CAD, which is abbreviation of Combined Annotation Dependent Depletion, is a tool for scoring the deleterious of a single nucleotide variants as well as insertions and deletions variants in the human genome. The insertions and deletions came later, so the first publication of CAD was restricted to SNVs, and after that they released another versions of insertions and deletions. CAD provides different types of scoring methods, but the highly used one is Phred-like score. This is a scaled score ranging from 1 to 99, and this based on the rank of each variant relative to all possible 8.6 billion substitutions in the human reference genome, which is an amazing tool that enables to provide. I could take any SNV or any insertion or deletion in the human genome, almost any, and ask myself, which is the prediction of the pathogenicity prediction of this substitution or variant. So Phred is a scaled score from 1 to 99. There is also a raw CAD score, which is less used, but for example, in Genix, we also provide this as information. The range is from 1 to 99 with the highest, so the higher, the more conserved and pathogenic prediction. As a thumb rule, this is not based on a specific publication or a paper or a proof, but it is considered that above 15 of a Phred score of CAD would be considered to be deleterious. And below that, we would suspect that this is a benign or not unpathogenic variant in the human genome. This is the UI of CADScore. This is how the tool looks like. You can download the entire data. It's a huge data set, but this is possible. And of course, in Genix, we provide this as an annotation source. But you could also look or search for a specific variant in the UI. So if you put a chromosome, you put the position, the genomic build, which just to remind you, this is very important to understand on which genomic build am I working on. You can see here that this is 37, which is AG19. And then the reference and the alternative nucleotides. And the alternative nucleotide is an optional field, because if you would not put anything here, you would just receive on the same position. If the reference is C, for example, you would get the Phred and the raw score for all different possible alternative nucleotides. So for example, in this position, the reference is a C. It could change to an A, a G, or a T. And this is the Phred score, which means that this is probably a harmless variant. So you can just go online, search for CAD, click on the SNV, and start to search variants just to see the prediction of CADScore. I would say that CADScore is one of the highly used tools today, and it tries to combine many different algorithms and many different features and characteristics of variants. The next prediction algorithm that I want to describe is SpliceAI. And SpliceAI is dedicated for splicing variants, not substitutions on encoding regions. And this is based on a deep neuronal network that predicts splice junction from an arbitrary pre-mRNA transcript sequence enabling precise prediction. Well, we can assess whether it's precise or not. But they claim to have a precise prediction of non-coding genetic variants that cause cryptic splicing. And we can see here are screenshots from the original paper. It's a great paper as well, recommended to have a further read on that. And then this is based on AI and machine learning algorithms to try to predict whether a new sequence in the splice region would cause a cryptic splicing. Synonymous and intronic mutations with predicted splice-altering consequence validated a high rate on RNA-seq in the original paper, which is a great advantage for this tool. And they are strongly deleterious in human population. So they did some kind of a validation on the RNA-seq level to show that indeed the abnormal splicing prediction correlates with the function on the RNA level. SpliceAI provides a score. This ranges from 0 to 1. This is the output, which is the probability of the position, specific position, being a splice acceptor or a splice donor, ranging from 1 to 0, from 0 to 1, as I said, with 1 being the most deleterious. SpliceAI also provides you information whether this specific variance is predicted to be splice acceptor or splice donor. So you not only get a score, but actually more information about the new sequence and the new prediction of SpliceAI in that region. What's very important to know about SpliceAI that we have SpliceAI for the entire human genome, for all non-coding regions, for all intron regions, which is very important when you want to analyze whole genome sequencing. And this is also important for CADScor. They're not restricted to a specific regions or only coding regions, for example. So both tools are very important when you want to analyze whole genome sequencing data. With that, I want to switch to allele frequencies, and the most important tool for allele frequencies would be NOMAD. And I'm going to take the next few slides to describe NOMAD thoroughly, but obviously I'm not going to cover everything. And there are many publications of NOMAD group, and I encourage you to read it. So NOMAD is a resource that was developed by an international group of investigators with the goal of aggregating and harmonizing both exomes and genome sequencing data from a wide variety of large-scale sequencing projects. And they provide a summary data that is publicly available for the scientific community. As an analyst for many years now, or almost a decade now, I could say that I could divide analysis before and after the release of NOMAD. NOMAD provided us high-quality data with high scale, which is also very important, and it provided it completely publicly available and freely, which is not the same in other programs like All of Us and the UK Biobank, which restricts the access to the data. There are a few version sets of NOMAD. The first one, the V2 data set, is only in Genomic Build 19. They did lift it over, so the information also could be found and queried in Build 38. And this provides, on the website, they provide 125 samples of exome sequencing, which is a great number, and additional 15,000 whole genome sequencing from unrelated individuals. So this is also important to know. Every individual in NOMAD would be the only one. There are no families or relatives, because obviously this would change or deviate the real allele frequency of the specific variant. The next version set was V3.1. This is only in Build 38, and it spans 76 genomes. So at the version number 3, they stopped sequencing whole exome sequencing. They looked ahead and they realized we have to provide more information on the genome level to discover novel genes, novel variants, novel diseases. And this is a large set of genomes that are in build 38. The V4 dataset, which was just recently, like 2 months ago, released, even less than that, includes data from a huge number of individuals, more than 800,000 total individuals. This is the largest data that is publicly available so far, and this release adds additional global diversities that includes, for example, 138 individuals from non-European genetic ancestry, which were the main samples so far. The V4 datasets contains also the UK Biobank inside, and I'm gonna describe it shortly. If we look here on this graph on the right side now, this is the sample size across the EXAC, which was the first name of NOMAD. So the EXAC was the first release, and then NOMAD took EXAC and used all the data and upgraded it with additional versions. But if we look on the EXAC, which was the version 1, NOMAD V2, V3, and V4, you can see the number of exomes and the number of genomes and the growth in every version that was released. And obviously, this is a great number of genetic data that is out there. So this is how the UI of NOMAD looks like. You can search for a gene, you can search a region or a variant, and there are some examples here. Very, very easy to search for a variant with just basic, like VCF structure. So you put the chromosome position, a reference variants, and the alternative variants, and you have here the link. And of course, you're welcome to go online, search for variants, search for genes, and have further information on them. One of the most important questions that we need to ask when we talk about a database of allele frequencies is what is the age and the sex distribution of the specific cohort. And this is important because when we are analyzing childhood onset severe disorders, it's different than analyzing adult onset disorders or some late neurological disorders for 60 plus age. And for that reason, we have the distribution plots here or graphs here, and you can see that the age of most individuals or most samples in NOMAD is about 55 to 60, 65 age of group. So there aren't many younger than 30. So whenever we look on an adult onset feature, let's say we want to analyze predisposition for cancer, we have to keep in mind that maybe the individual that was sampled in NOMAD at the age of 35, for example, was healthy, but then maybe 5 years, 10 years later, the same individual could have developed a specific condition. So the fact that we have an individual carrying a specific variant in NOMAD does not mean that it is a perfectly healthy individual. So we always have to keep in mind the limitation, as I said, for every data source. Now on the right side, this is the age distribution of genomes, and we do see that there are more genomes of 30 and less samples. In terms of the sex, so this goes about half females and males. Now the next most important characteristic of these samples would be the ancestry group that they belong to. Obviously, there are different ethnical backgrounds and different ancestries worldwide, and we want to be able at least to capture all of them in such a database that is publicly available and everyone is annotating against. And while the first versions were mainly European ancestries, and we can still see that in V4, there is a great effort to try to increase the diversity of this database. In NOMAD 4, or even before that, in NOMAD V2, they were, and we can see them here in orange-like color, there were 5,000 Ashkenazi Jewish individuals, which made much easier the analysis of a Jewish individual from an Ashkenazi ancestry. There are also African, Middle Eastern, and South Asian and East Asian in NOMAD v2, but there are many populations and ancestries that are completely missing from NOMAD, and this is still the situation. For example, the Middle East, and in one of their publications, they stated that the Middle East populations are completely absent, or almost completely absent from NOMAD, and there is an effort to change that and to provide much greater diversity of sequencing samples for reference and for annotation sources. So this is NOMAD, a great data source that not only provides us allele frequencies, aggregated allele frequencies, it also provides us a constrained matrix per gene. So once they aggregated a lot of data, they realized they could do a lot of statistical measurements using or based on this data. And one of the publications of NOMAD was about constrained matrices. And here I've put the same gene as we saw before, AGBL1, which is responsible for corneal dystrophy and an autosomal dominant inheritance model. And I put it into NOMAD, and I've provided a screenshot from the right upper panel, which is the constrained matrix of this gene. And I want to further describe the constrained matrix that is provided by NOMAD. So we can see here on the left side the category of the specific variant, and this is on a gene level. Again, just a quick reminder, we have to know whether this annotation is on the gene level or on the variant level. So if I put a gene, I would receive all the variants that were found in the sequencing of the samples in NOMAD version sets. And then they would provide them, or they would categorize them into 3: synonymous variant, missense variants, and loss of function variants, which are the four variant effects that I've described. And for each and every category, variant category, now they can calculate a constrained matrices, and these would help us learn more about the gene. So if I start here in the expected SNVs, these are the expected variant counts that were predicted using a depth-corrected probability of mutation for each gene. So based on the length of the gene, there is a predicted number of substitutions, or SNVs, that is predicted to occur. And more details could be found in the NOMAD flagship paper. You're welcome to go and read. There is also a link from their website. And they just provide here an additional note that the expected variance counts for bases with a median depth of above 1 were removed. So anything that was less than 1 was removed from the total. Okay, so this is the expected number of SNVs for this gene based on its length, and then there is the observed SNVs. So this includes a single nucleotide change that occurred in the canonical transcript that were found at a frequency of greater than 0.1, and here is again at a depth coverage of more than 1x at the specific variant, and we talked in the previous class about the median depth of this variant. And if the frequency was more than 0.1, less than 0.1, and it passed all filters, and at the site with the median depth of above 1, the counts represent the number of unique variants and not the allele count of these variants. So overall, we are counting how many variants, how many SNVs variants are found in this specific gene, the AGBL1, that are synonymous in the first category, that are missense in the second category, or that are loss of function in the third category. And this is the actual number, the observed number of variants when the median depth was above 1, because of course, if the region is not covered, then we cannot assess the number of variants in this region. And of course, they count unique number of variants without looking on the actual allele count or the frequency of every variant. And now by comparing the expected SNVs and the observed SNVs, Now they can talk about the constraint matrices and the parameters that characterize these genes. And this is divided into z-score, z-score, and PLI. Now, the z-score designates the deviation of observed counts from the expected number. When you have a positive z-scores, that indicates an increased constraints, which means intolerance to the variation, and therefore that the transcript had fewer variants than expected. Now, I've just said transcript, and this is very important to understand that the entire analysis of the constraint matrices go by transcript. So we choose a specific transcript for every gene. There is a single transcript, and this could be usually is the main select transcript, which is the commonly found transcript for the specific gene. If there is no main transcript, because there are main transcript, main is a defined transcript, a specific transcript for each gene. If there is no main transcript that is known for a specific gene, then they take another transcript, which usually is the canonical transcript or the longest transcript. Now, positive z-scores indicates, as I said, intolerance to variation, and negative z-scores that were given to transcript that had more variance than expected, and this is exactly the opposite, which means there are endless number of variations and there is no constraints on the variation in this region. The PLI and the PLI measurement is only relevant for PLOF, for loss of function, predicted loss of function variant. And this is the probability of being loss of function intolerant. If I have a high PLI, let's say its PLI ranges from 1 to 0. For high PLI of 1 genes, this means that this gene is completely intolerant for loss of function variations. If I have a low PLI, like here, which is the PLI of 0, then that means that there are many samples with loss of function variant on an heterozygous zygosity. And this is completely okay with the gene, which means this would not harm the basic function of the gene. And we know that almost all enzymes when we have 2 copies of them, and that means that you can lose 1 copy, and it is okay that there is only a single copy that is functioning and provide the full functionality of the enzyme. These genes, the enzymes, would usually, almost always, would have a PLI of 0. Another measurement for predicted loss of function variant is the observed versus expected. They call it OE. And this is the ratio of the observed versus expected number of loss of function variants for all bases of sufficiently covered in the main select or canonical. So as I said, they took the main select or the canonical one, and if there is no canonical one, then they choose another non-select or non-canonical transcript for each gene. The observed versus expected is considered to be better or more accurate than the PLI, and overall, most labs would take PLI of above 0.9 or high chances of being a dominant inheritance gene. And with that threshold, if you want to convert that threshold to OE, then you have to take a less than 0.35 value of a OE, which is the same as a PLI of above 0.9. So the observed versus expected is more accurate than the PLI. They provide many claims to that on their website and in their publications. And we provide in GenX both measurement, as I will show in a second. But just to say, the OE, which is described here, if it's the less the score is, the more restricted the gene is for loss of function variance. In the GenX, this comes from the GenX platform, you would see the PLI and the OE measurement as coming from GNOMED. Next, and I don't have a lot of time, so I'll try to rush this one. I want to describe ClinGen. ClinGen is a very important data source that curates genes and their dosage sensitivity. So we talked about PLI, which was a technical or a computational algorithms that reviewed variance expected versus observed variance by specific categories, synonymous, missense, and loss of function variance. And by a computational algorithms, they provided us scores that helps us understand whether a gene is predicted to cause disease in an autosomal recessive manner or autosomal dominant manner. But the ClinGen is a committee that is dedicated to provide highly curated and validated information on genes based on publications, on known diseases, so not on a computational prediction. And this is a valuable resource for deciphering how changing gene copy number affects health. It provides us information on haploinsufficiency score, HI, and triple sensitivity. Haploinsufficiency is almost like the PLI, again, not based on a computational evidence, but based on a literature review that actually provides you information regarding is this gene is an haploinsufficiency gene, which means can this gene function in one allele, or the gene requires the function of both alleles to function properly. That also means that if a gene is haploinsufficient, then it would lead to a dominant disorder. Now, triple sensitivity is this gene is sensitive to have 3 copies of the same gene or not. Now, Genix leverages this information from ClinGen expertise, and we provide the dosage sensitivity data for both individual genes and also specific genomic regions. We can see here a screenshot from ClinGen. We have the HI score and the TS score, again, haploinsufficiency and triple sensitivity. The scores of HI would go 0, 1, 3, 40. If you go online, you would see the number of the score and then the actual outcome of the score. For example, 3 here would be sufficient evidence for being an haploinsufficiency gene. So as I said, in ClinGen, there are gene level analysis and there are region analysis. ClinGen evaluates genes for haploinsufficiency and triple sensitivity, and we obtain the same lists of, for example, these are haploinsufficiency scores from ClinGen. We provide the same information in Genix on the gene level, but there is also a region level analysis because sometimes you have a missing an entire chunk of the DNA, as we know from microdeletion syndromes or microduplication syndromes. And we try to take this information from ClinGen, and you can see here on the Genix screenshot here at the bottom part, ClinGen haplo and ClinGen triplo, to show that specific regions, when there is a large deletion, there is sufficient evidence for dosage pathogenicity. This should help you analyze the regions mainly for copy number variations. Before I go to the clinical case study that I wanted to show you, this is just a screenshot of all the annotation sources that are in Genix, and there are many other levels of gene levels and region levels, annotation sources. But what's important in this slide is to understand version set, for example. Every data source that we use has a version set. For example, if I want to use ClinVar annotation, ClinVar updates daily on a daily basis. On all platforms, on all annotation sources or processes, you would take a screenshot of ClinVar at a specific date and you would use it as an annotation to your data. And obviously, a year later, a month later, a day later, this would not be up to date. So it is very important when you analyze a case and when you issue a report that you would provide the version set of ClinVar that you worked with. Same for CADScore, same for OMIM, same for all annotation sources, because the version stats is your insurance certificates to say, I was up to date at the date of the report. So you need to know on which version you're working on. And for that reason, if you click on the version set in Genix, you would receive all the dates and the version numbers of all the annotation sources. Very important feature to have a version set control and to provide all the version sets inside every report you issue. With that, I want to start a case report, and I'm going to go fast and describe this. So we can see here on the right side the pedigree. This was a male patient with the following phenotypes: speech disorder, recurrent infections, dystonia, and developmental delay. And this was the second child to healthy parents and one healthy sibling, and the mother was pregnant with a fetus. We can see it here. And we conducted a trio exome sequencing. It was a rush one, was an urgent one because the mother was pregnant. And luckily, this is the Genix platform. I think this is the first time that I provide a screenshot of the variant view. So we can see here, every row is a variant with all the annotation sources gathered in columns, in different columns here. And the phenotyper, which correlates between genes and phenotypes, rank and sort based on the ranking every variant to the correlation of the specific phenotypes. And we can see that the first row was a variant in a gene named PDHA1. What we can also see, and let's go to the next slide, after we get this visualization, in the next class, we would try to solve cases together. I just want to give you an overview of how do we know that this is the correct diagnosis? What do we assess? So we look on ClinVar and we can see right away that this is a likely pathogenic variant by ClinVar. If you would click on that, you would get the ClinVar pop-up with all the information from ClinGen, if this is a variant that was curated in ClinGen, and all the different submissions in ClinVar. Now, these are not single submissions. These are RCVs gathered or grouped based on the condition, as I've described earlier in this class. I can also look on the phenotyper, and I see that there is a very high correlation. If I click on the score, I would see all the matching phenotypes with all the lists of evidences coming from the literature, from the data sources that a phenotyper uses. Next, I could look on the gene. If I click on the gene, this would be the gene pop-up, and there is a phenotype, inheritance, and the OMIM-ID. There is a lot of other information on the gene level, like the constraint matrices that we have described from NAMAD. And then there is also diseases from Decipher and summary of the pathogenic and likely pathogenic SNVs from ClinVar for this gene based on the effect of the variants. Because if you have, for example, missense variants, you want to know how many missense variants were reported to be pathogenic before for this gene to understand the pathogenesis or the pathomechanism of this specific gene. Is it a gain of function? Is it a loss of function mechanism? Next, I could also try to assess the inheritance pattern. So I see that my patient is hemizygous for this variant. It's an X-linked variant, and it's a male patient, as I've described. And the variant is inherited from the mother, which is a carrier. So this makes sense in terms of inheritance. I could also see that this gene is known to be X-linked gene, so that is also fit with what I see here for this as a diagnosis for this patient. And when I look on NAMAD on the maximum allele frequency here, I see this is empty because this variant is absent from NAMAD, which also makes sense. Now, this is the report that I've issued for this patient. We described the SNV with all the standardized method of nucleotides and protein, which AGVSC, AGVSP. I provide the allele frequency from NAMAD and the dbSNP number, and of course the position and the transcript that I've assessed. And I can also provide a segregation table based on the trio analysis that we conducted here. And I can give the information about each and every individual or associated samples. It could be a family of 3 siblings. It could be a trio with a proband and 2 parents, etc. And I would provide the segregation analysis table in the report. And then obviously I should also provide a description of the known phenotype from OMIM and the known information from ClinVar in the report. With that, I want to thank you for listening, and I'm open for questions, myself and Dr. Ali Tavish.
Thank you so much, Dr. Hagar and Ali, for being here. And Hagar, that was really amazing, really informative presentation. Thank you so much. Let me just find Hagar quickly.
I think, Jade, we don't have much time, so we have to be quick.
Okay, go for it. I mean, Hagar, are you ready to get going?
Okay, let me. There was one question about gain of function. How do we predict gain of function? Right? Somebody was asking. So let me explain. If you are doing variant filtering and you have a novel variant, and there is no publication, there is no annotations, this variant probably end up as a VUS, as a variant of uncertain significance. Unless a variant has been published as a mechanism, a gain of function mechanisms, we can handle that accordingly. There is publications that Ravel can predict gain of function, but these predictions are not very reliable. There are some new AI tools which help in the prediction of gain of function mechanisms. We can get help from those, but they are not very reliable. So just to answer, the most reliable is the publications, the published data. There was another question regarding to using the internal data sets. It has to be published data set that we can use in our variant annotations, variant filtering, report writing. Okay. I think there was another question on.
Hi, Ali, let me jump here. Sorry. So one of the questions that were posted in the chat is about computational filtering, which was at the beginning of the session. And the question was, how does one protect specific alterations like variant of unknown significance with the computational filtering that takes place at the beginning? And I think it's a very fundamental and important question, so I do want to take a few seconds to answer. So this is a great concern. It's a very important concern. When we run into or when we initiate the computational filtering, we should always suspect that we might filter out the pathogenic variant. We have to make sure that we are not doing that, but obviously it could be a variant of unknown significance, and these would be filtered out based on, for example, allele frequencies. So we all know that there could be a situation in which the pathogenic variant or the causing variant is highly frequent, but you would remove high allele frequency variants, and with that removal of variants, you would also remove your causing variant. So what we try to do is we try to protect certain variants that are known to be pathogenic in advance, because there is a closed list of variants that we can secure to be protected from the filtering. Another approach would be to use ClinVar. So if you have a pathogenic or likely pathogenic by ClinVar, you would flag it as always show likely pathogenic by ClinVar. This is the approach we do in GenX. So whatever filtering approach you are doing, if it has a registry in ClinVar for pathogenic, you would still see the variant. I think we are out of time, and there are tons of other questions. But thank you for this fruitful discussion. I think we'll try to address them maybe in the next class. So we'll start maybe with these questions.
It's a great idea.
Sounds good?